Hello everybody and welcome to Brock University. My name is Belinda Kemp. I recognise some faces but not everybody. I'm the new Oenologist here at CCOVI at Brock University. And first of all I want to thank our sponsors who obviously are CCOVI and Vines to Vintages as well and Laffort. I'm going to introduce you to Dr. Peter Salamone. I've been practising his surname all lunchtime. Peter's the technical manager at Laffort, North America. He has broad experience in industrial biotechnology and within the wine industry within production settings such as Diageo, Chateau and Estate wines. He's also going to tell you a little bit more about his experience, but he's also carried out work in molecular biosciences, so things like microbiology, enzymology and genetics. So I'm going to pass you over to Peter for the next hour, and when he's finished in about an hour or so, we're going to have a question and answer session. So I'd like you to join me in welcoming Peter Salamone.
Thanks, Belinda. And thanks everyone for taking time out of your day today to come out for the presentation. I hope that at the end of the day there'll be some interesting things you can take away and say that it was worth your time. That's really my goal at the end of each of these kinds of presentations, is that you feel like it was worthwhile at the end of the day. How many people were here earlier this year when I gave a talk on Surly Science? Perfect, whole new audience. Okay. So this is kind of a different talk today. It's everything is included in the title. It's a really broad talk, so I didn't really know how to give it a title. So I just throw this up there. We'll just go through topics a little bit at a time. As Belinda said, we'll have lots of time at the end for an in-depth discussion. But if at any point in time as I'm on any of these topics, a question comes to mind, feel free to raise your hand. We'll stop. We'll chat about it right then while it's fresh in everyone's mind. I'll give you a little more background on myself, because who doesn't like to talk about themselves? Like Belinda said, I've got a lot of experience in molecular biosciences. I was a biotech guy long before I was a wine guy. I did my PhD at the IBC at Washington State, which is basically a molecular plant physiology department, which that's about as close as I can get to talking about some of the viticultural things that I do speak about. I know some plant physiology and secondary metabolism, and I learn from viticultures every time that I get to speak to an audience, and there's some vit folks there. But the Washington State experience was really great for me, and it enabled me to do what I did moving into another field. I got recruited by a small family winery in the Central Valley of California, Ernest and Julie O'Gala. So that's where I started out. I worked for Gal as the lead biochemist from the technology and quality department. So I had my hands in a lot of different things and got to deal with a lot of different winemakers and hear what challenges were across the board and try to find solutions. And so, from there I've done a number of different things. I went back to biotech and worked for an enzyme company for a while. And then I was back in the wine business with Diageo, Chateau and Estates, which was a very different business model than Gallo. A lot of different smaller wineries, still very big wineries like BV and Sterling and Etna Valley Vineyards and Chalone. They're fairly large, but not as big as Gallo. So it was a completely different paradigm of how business was done. And again, I'm sure everybody here doesn't come from wineries that process as much fruit as a BV or a Sterling as well. So again, it's the processes that you use are very different. So, and with the things we talk about, these principles, they change with scale. So if there's things that don't seem to make sense to you based on your experience and what you've done, raise your hand and bring it up, and let's talk through how these principles apply actually to what you're dealing with and what you have to do in your winemaking. Okay. So with the scope of the talk today, we're talking about aromatic white wines and flavor aroma molecules and oxidation and extraction and preservation and all this. I'd like to start at the very beginning and say, what's the deal, what's the molecular basis for aroma and flavor? I've inserted an awful lot of fundamental science in this talk, but it's not all fundamental science. Everything I do that's a slide that you say, gee, Pete, that's pretty involved cell biology. Why are you telling us that? It's got not much to do with wine. It'll come back to wine. When we start talking about the principles involved in wine, it helps to understand a little bit more of the systems that we're working in. So really, how do we perceive these molecules, right? This is just as important. If you want to know what state you want to keep them in, you have to know what state are they important for the sensory perception. So you've got taste, sensation, tactile sensation, and smell. And these are all very different in humans. You can see I've got an illustration of a taste bud here. And there's only 5 things that you actually taste. There's only 5 different flavors that are perceived on your taste buds. And these are those guys. And they're contributed by acids, sugars, peptides, tannins more so with tactile, I believe. But there are some tannins that can give bitter taste sensations. So I included those in that category. As far as tactile, this was an interesting talk. I just finished a technical series that I did in California called Chasing Mouthfeel from Vine to Glass. And I had 4 invited speakers. One was a viticulturist and a chemist, and then we had Anita Oberholster who helped develop the mouthfeel wheel, and a researcher from Italy who worked on salivary protein-tannin interactions. And one of the things that came out of that talk is the chemist, everybody pretty much spoke about the interaction with tannins and salivary proteins and creating the sense of astringency. And I challenged that. I said, it doesn't make sense to me. The tannin interaction with the protein doesn't cause astringency. It's a precipitation event, the tannin binds it, but that's not astringency. And finally, it came out, there's mechanoreceptors in your mouth that when these proteins are stripped away from them, the sensation of the tissues rubbing against each other, that's what you perceive as astringency. So it's really not the interaction with the protein, it's a subsequent feeling in your tissues that's mediated through that interaction. So again, think things through a little bit, ask questions, and there's answers, and it's not the assumptions that get made in what to do in winemaking because of instances like just a small instance like that. How do I moderate that? Well, it's more about shielding the mechanoreceptors than it is about blocking that specific reaction, because the real sensation comes from the mechanoreceptors, not from the protein-tannin interaction. So again, all right, so we've got tactile sensations. Kind of went down the rabbit hole on a little bit. This is an illustration of a salivary protein. This is not an exact salivary protein, but they're extended. Most proteins are globular. The amino acid chain folds in on itself, and they're globular in nature. But salivary proteins are linear like this, and they're made that way because they're supposed to interact with a lot of different molecules. Smell, I always like to talk about this because a lot of the things that are involved in smell, you think about them as flavors, right? You taste a raspberry. But you don't taste a raspberry. This is all you can taste. Okay, you smell those things, and this is where it gets into the sensory science kind of things because it's perception. Okay, it's perception. It's not a single molecule giving you this, but it's a number of inputs, including the flavors that are on your tongue, the smells that come up. This is an illustration of kind of sliced through a human head. Here's your tongue down here, here's your throat, and here's your nasal cavity. Here's your olfactory gland, and these all have neurological connections up to your brain. And in part of your brain where all these connections come together is your perception. Because it's not a single input, it's multiple inputs, which is why it's really cool. When you talk about wine flavor, right, you smell a wine and you say, oh, lychee, or grapefruit, or passion fruit, or raspberry, or whatever, you're associating it with something else. You're not describing that specific individual perception. You're associating it. And it's because it's complex with all the molecules that come in there. What we're going to focus on mostly today with these, with the aromatic white wines and what we're going to talk about is mostly these compounds. These volatile compounds that are perceived at your olfactory gland and in cooperation with your taste buds and your tactile sensations give you the perception of wine and what it is. So, speaking of starting at the beginning, this is a grape. So, I think everybody knows what a grape is, and everybody knows their skin and pulp and seeds. So, the point I like to make with this slide is that the aromas that come from grapes, 80% of those aromas are in the skin. Only 20% are in the pulp, and there's none in the seeds. Okay? Now, and when we talk about grape and we talk about wine, we say, well, if the flavors really come from the aromas and 80% is here, then this is not that important. But this is all your sugars, it's all your acids. It's what creates the balance in what wine is. We're not talking about phenolics today. So in a little bit more detail, you can see the classes of compounds that are in the skin. These are the guys that make up these aroma molecules, terpenes, norisoprenoids, thiols. We're going to talk a little bit more about those molecules and where they come from. The biochemistry inside the plant cell that creates these, the cell physiology as to where these molecules are stored and moved within the cells. And you're like, Pete, I didn't come to science class. I want to know about winemaking. Well, that all comes back to, we want to extract these. We want these out. We want them in our must. We want to ferment them. We want them to be modified or preserved. They're a big part of what makes a quality wine. So the more you know about the foundation of these, the more you can strategize appropriately with your winemaking to bring these flavors forward in your wines. Okay. And then in the pulp, there's of course the acids and sugars. All right, so small number are represented here. This is only a few. I've got longer lists. We'll have a long test on it afterwards. But so there's a good deal of these, and I'll get into why there's so many as well. And then volatile compounds, such as these class of molecules, they're stored as conjugates, sugar, amino acid, organic acid conjugates in the cells. And basically, once these things are conjugated to a sugar or an amino acid or an organic acid, they're not volatile. Okay, they're soluble at that point. So if they're not volatile, how are you going to experience them at your olfactory gland? As you take that wine into your mouth, compounds have to become volatile to go up through your nasal passage to your olfactory, so you sense that. Couple it with the flavors from your tongue and the tactile sensation and say, wow, this wine is really complex. This wine is really wonderful. I get this, I get that, I get the other. Otherwise, it's like drinking sugar water if you don't have that sensation. But if all your flavor aroma molecules are conjugated, how do they get up? How do they become volatile? Through the crushing process and subsequent cleavage by grape, yeast, and/or industrial enzymes during the winemaking process, or through cellaring and storage at wine pH in an alcohol environment, these molecules will cleave those conjugates off and become volatile. So that's your aging, your cellaring, your maturation of your wine flavor. And there's ways to impact that through winemaking practices as well that we'll talk about. Okay, varietal character in white wines, including, I list a bunch of wines up here, some of which would be categorized as aromatic and some maybe not. But the fact that these classes of molecules we're talking about, they contribute to the varietal flavor. So I've listed a bunch of different white wines here. Just for the sake of saying so, how many people here grow Sauvignon Blanc? Okay, a few Riesling, about the same amount. Gewürztraminer, around the same amount. Traminette, nobody does that hybrid here. Okay, interesting. Muscat, growing Muscat here, just one. Okay, Chardonnay. The weed grows over there. Okay. All right. So the character in these many different varietals, and they're all different. I think that we could have glasses lined up, and I know it would be a stretch for me to say I could pick out which varietal is which, but I can darn sure tell that they're different. It's determined by the presence, whether it's there or not, and the concentration of compounds from these chemical families: norisopyrenoids, monoterpenes, thiols, chemical and metabolic organic molecules, fermentation esters, things of that sort. So here's where it gets bad. So this is a picture of a cell. This is the outer membrane. This is a cell. Okay. I'd like to make a few quick points about this. Just that these inside of a cell, whether it's a yeast or whether it's a grape cell, these are called eukaryotic cells. Okay. They're higher organismal cells. Bacteria cells are a single cell. They have a single membrane. They don't have any of these things inside of them. Okay, they've got a genome and they've got some cytoplasm, but they don't have these compartments. Okay, and I want to make this is a pretty cool biological point. Each of these things, like a plastid or a vacuole or a mitochondria, peroxisome, and even the nucleus where the genome is, these are all lipid bilayers. Just like the outer membrane is a lipid bilayer. It's a barrier. Water can't pass freely through it. It's very hydrophobic. And there's barriers inside of eukaryotic cells as well. This is compartmentation. Okay? This allows eukaryotic cells to do chemistries that simpler cells can't do. Because if it's one environment, there's only a certain amount of reactions that can take place. Okay? And you say, Pete, well, how do I care about this? This compartmentation and segregation enables biochemistry to generate a lot of diverse molecules, like the norisopyrenoid family, the monoterpene family, the thiol family. Okay? The more diversity that the plant can generate, the better it is for us as winemakers in terms of generating that kind of rich bouquet that we like in wines. Okay? So when you talk about where these things come from, I don't want to just say plant cells make this. Lots of people say that. I like to give you a little bit more. plant cells make this, and they make these things in these different compartments, and they get stored in vacuoles. So the other point I like to make about this, another cell biology point, is there's not a vacuole in a cell. Okay, I've got an electron micrograph later. You'll see some different. There's little ones, and the plants are like all biological creatures. They're not stupid. They're really, really smart. The way they've set themselves up is really smart. These vacuoles have different cell or membrane surface components, specific transporters that bring into those vacuoles what's meant to be in there and keeps out things that are meant to be out. Okay? They're pretty smart. The biosynthetic enzymes for some of these flavor and aroma molecules sit on the surface, embedded in this lipid bilayer. They make the molecules out here, and then they're transported inside. The last step before transport is usually conjugation. Add a molecule onto it, that molecule becomes the target of the transporter to pull it inside. Why would plants do that? Segregation, concentration, stabilization, all of those things are important. These are the molecules we want in our wine. This is our flavor aroma. This is what a plant's doing to make all those and keep them. Okay? So, norisoprenoids. These are an interesting family. You can see that they've got very diverse structures, rings and double bonds and all that stuff. The structure isn't what's important. You may have heard some of these compounds, beta-demescanone, ionone. These are, when you read papers about flavor aroma, these are names that get thrown out there. This is like the major flavor component in red wines. So it's important to these, but the other thing, here's one, TDN, also listed as its sensory threshold level. So you can see all the way down at nanogram levels or up at microgram levels or hundreds of microgram levels. The sensory threshold for human detection of these molecules and the different sense that they give varies quite a bit. So the concentration of these varies quite a bit. This is actually kind of a major pathway. There's a lot of this, plus it's a low threshold. So that's why it's so predominant in red wines as a flavor. And it's not, like I said, there's lots of different molecules that give the characteristic flavor, but this is a major one. Also, just to note the structures on this, they all look fairly similar, but they're all very different. And the thing I like to point out here is these kinds of molecules like this or like this hydroxyl, these are the targets for oxidation. Okay, that's what those particular functional groups, when these molecules are not conjugated to a sugar or whatever, when they're volatile and they go up against your olfactory gland, your olfactory is very complex, but it's nothing compared to a dog's. Okay? Dogs can sense millions of times more molecules than we can. Our sense of smell is nothing compared to theirs. But still, with all of these diverse molecules, thousands of them, there's a different receptor that binds them. Okay? And if these are modified in even a slight way, it doesn't bind that receptor, and that sensation that it brings and that creates that perception of the flavor in your brain doesn't work. We'll see when I look at oxidation, you'll see a difference between this versus a aldehydic group at this one location. Makes all the difference in the world as to whether your wine smells fresh and fruity or aldehydic and old. And we'll just mention too these kinds of things, kerosene and petrol. As far as like Riesling flavors, it's got nothing to do with the molecules that are kerosene or petrol, but the perception that it gives when these molecules are sensed on your olfactory. That's why you get those kinds of descriptors for Riesling flavors or even yours. Okay, so where do these norisoprenoids come from? They come from a class of molecules called carotenoids. You've probably heard about carotenoids. You hear a lot about vitamin A and what it does for your vision. Everybody's heard of chlorophyll, right? But these molecules, this family of molecules, they're not, chlorophyll actually takes the electron energy and transfers it through a cytochrome chain. It makes energy for the cells, basically. But there's a lot of other molecules that look like this, that surround chlorophyll in those tissues, that capture light energy and feed it to the chlorophyll so it can then interact and make energies. This is a pretty big class of molecules, and it's in green tissue, right? Green tissue is green because there's chlorophyll in it, and it's got a characteristic color. These guys don't always have colors in their native state. The chlorophyll overpowers them, so you see green, but these guys are all in there. What color are your grapes before veraison? Green? Even red grapes are green before veraison, right? So these molecules are in there. What happens during senescence or maturation, these things will break down. All All right. Some of it is done enzymatically, and some of it is just done through senescence of the tissues and normal degradation of molecules. Plants are amazing biochemically. They don't waste anything. They don't just take stuff and throw it away. That's why the plants turn brown and all before they fall off, because they're senescing. All the good stuff they're breaking down, shipping into the phloem, using it in other tissues, and then they get rid of the framework that it was in. So they reuse a lot. That's what goes on in the vacuole of the cell, the main vacuole. Lots of degradation, lots of proteases. Plants, so cells put stuff in there, it gets broken down, the parts come back out, and they get reused again in different reactions. It's the same with this. The plant breaks this down during senescence, during maturation. These molecules, this diversity of molecules that plants make, why would they make all of these things? Thousands of these different molecules. Plants are warriors. They're in one place, and they fight their whole life for their existence in one place. They can't run away, they can't move, they can't brush things away. They fight biochemically. Okay? This is the plant's immune system. Okay? It's its communication system. It interacts with its environment through biochemistry. It accumulates these molecules as attractants for beneficial organisms around it, or repellents for organisms that are not good for it, disease organisms like fungi or grazing organisms or insects that eat them. They make molecules that repel these things. And they also communicate with each other through this kind of molecule, right? Like pheromone kind of communications. Lots of these molecules fall into that class as well. So, plants don't make this so they smell really nice or help us make nice wine. They use this, it's a pretty diverse system that they use. We only need a few of these guys to help make our wine good, but this is where they come from. These precursor molecules that are doing a completely different job. And then when their usefulness is finished, they become breakdown products and become a completely different class of molecules. Here's beta-demescanone, here's iodone. So, okay, what else you got, Pete? All right, here's just another example of a breakdown product. You got neoxanthin. And it breaks down and you get this grasshopper ketone. I love this grasshopper ketone. What a great name. Okay, and then through another bioconversion through an enzyme, it turns into this compound, then into this compound, and then beta-demescanone. So you can see it's a hydrolytic event, and then it's 1, 2, 3 conversions to get to this compound. Do you think this is random? It's not random at all. It's part of the plan. There's an enzyme, and for every enzyme, there's a specific gene that's regulated 3, 4, 5 different ways. Okay? Plants are pretty impressive with their biochemistry. All right, so just to say, it's just another mechanism. You can see degradation, biochemical conversion, and this is just one of thousands of different molecules. All right, enough of the norisopyroids. How about terpenes? Where do they come from? You've all heard of terpenes, right? Heard that terpenic character to a wine? Anybody ever said that or heard that used as a descriptor before? What did it mean to you? What kind of flavors did you associate with that descriptor? Okay, we'll see on the next page. I've got a lot of different flavor descriptors for different monoterpenes. But terpenes occur in all plants, represent the largest class of secondary metabolites. Over 22,000 compounds have been described. That's just what we've described. I'll guarantee you there's more. So here's your basic unit. The simplest unit is an isoprene unit. It's a 5-carbon unit. It's illustrated right here. Okay? They're synthesized in a plastid. Okay? Compartmentalization of biochemistry. Colorless plastid, similar to the chloroplast, but again, its surface membrane is very different. It's got different transporters in it. It does different chemistry inside and transports different things out. This is a 7-step catalyzed reaction to get to this point. And then monoterpenes are produced from isoprene subunits. So you can see isoprene, 2 of these together is called a monoterpene. So it's a 10-carbon unit. It's a monoterpene. Here's an example of linalool. You may have heard of this before. Larger than this, if you add another isoprenoid unit, it's a sesquiterpene. We don't need to worry about these because these aren't really compounds that are associated with wine and wine flavor aromas. But just to let you know, there's a bigger world out there than what's going on in these plants for terpenes and wine, and monoterpenes and the flavors they bring. There's all these other classes of higher level molecules, and these guys especially are used in a lot of communication, and a lot of these have insecticidal or repellent features to them, these larger isoprenoids. But what's important to us? Okay, monoterpenes. A lot of these guys, linalool, nerol, terpenol, geraniol, citronellol, nerol, geraniol, These guys, these are the ones that when you read articles where people talk about the terpene-driven varietals like a Gewürztraminer, okay? These are the kinds of compounds that they look at and they'll measure comparatively this from this vineyard, this from that, identified it like this and like that. And sometimes you'll see these things measured as a means of describing the aromatic content of the wine. Well, here's what they are. And they're pretty cool molecules. You can see they're all a little bit different, but the important thing here I want to show you is this terminal hydroxyl, this is carboxyl hydroxyl. But these are the guys, that's where the conjugation occurs. Okay? That's where the plants will add on a sugar molecule or an organic acid or an amino acid like that to protect these, to make them more soluble, and to target them where they need to go. Again, big class of molecules. So here, just as a little bit of a dendrogram description of these different monoterpene molecules, you can see that down here, these guys will group around the Gewürztraminer descriptors. This with the Treminette that we spoke about, the hybrid, and the Riesling. So you can see that there may be some overlap a little bit with like the terpenol and the terpenol, but for the most part, you can separate these in terms of which molecules are there. This is just a very small sampling. But the entirety of what's there gives the varietal characteristics. Okay, and you can see these monoterpenes have descriptors like sweet and fruity and different specific things like orange or citrus. There's floral descriptors, lavender, rose. I saw hyacinth on there. So there's a lot of citrus, waxy. This one probably gets oxidized very easily if it's got a waxy type aroma. Woody, peach, anise. That's licorice or anise. So these things do an awful lot of what we like in terms of positive flavor compounds. All right, real quick, because nobody really said they do anything with Treminette, but you can see that in terms of total amounts of monoterpenes, the Treminette has significantly more than either Gewürz or Riesling. So let's see. Yeah, okay. So over 50 identified in grape and wine. Again, in plants, 22,000 overall. In grape and wine, we've only gone so far as to identify 50. So there's a lot more. Treminette has about 2 times more, at least double Riesling. And the Riesling fruit profile is really dominated by norisoternoids. So these are terpenes, and it's got a pretty decent level, almost as much as Riesling, but the norisoternoid family really dominates the profile in Riesling. So the third group of compounds, the major group of compounds for flavor aroma, thiols. Okay, I did this presentation over at Cornell yesterday, and Gavin Sachs did a whole presentation on thiols. So I didn't include a whole lot in here. And basically what I talk about when I do this is just these 3 main, I think of 3, MMP, 3MH, 3MHA. These are the main thiols from Sauvignon Blanc. These guys give the characteristic flavors of passion fruit, the box tree, grassy flavor, the grapefruit character. These 3, this is actually the box tree grassy character. This is the grapefruit. This is the passion fruit. So you can see they're made as precursors, and they're conjugated, and they're not volatilized. You don't really smell them until they've been released from the conjugate. But you can see that another point I like to make about thiols is in juice, you can see, here's a profile. Each one of these bars represents a different thiol compound, and the height represents the approximate concentration. Okay? Here's what you would see in juice. After fermentation, here's what you would see in the same wine. Yeast have a tremendous impact on the volatile thiol composition of wines. Okay? They make a number of compounds, they modify a number of compounds that come in the must, and they also have enzymes that release the conjugates from some of the major thiol compounds in grape must. So very important for thiols and the yeast that you use. Okay, so we've talked about modification a little bit. I'm not going to get down the rabbit hole too much on this, but here's this name, glycosyltransferase. Glycosyltransferase. Okay? Gly means sugar, any sugar. If that was glue, it would mean glucose. It's a specific glucose transferase. But this glycosyltransferase transfers numerous sugars, different sugars. It's kind of a promiscuous enzyme, the kind I like. I like enzymes that do a lot of things. Okay, so why? Why have this enzyme? Why add sugars on to our compounds? Why add 2 sugars on? This is the way these compounds come. Usually they're diglycosylated, like this. This sugar is almost always glucose. And this sugar can be one of 4. It can be glucose, or ribose, or amylose, or abiose. Okay? Pete, I don't care about these individual sugars. What's the big deal? Come on, I just want to make wine. Well, you need an enzyme to cleave this off and an enzyme to cleave this off. There's an enzyme that cleaves them both off. It's a yeast enzyme. It's really, really low concentration and doesn't last very long in the fermentation. So it's there, but it's not going to help you release these aromas, especially not long term. Okay? So, I said you need an enzyme to cleave these bonds. You don't need an enzyme. Acid, time, heat, higher alcohol, organic solvents, all these things will contribute to these bonds breaking. Okay? Enzymes catalyze reactions so they happen faster. Enzymes work so you can control what's going on instead of waiting and seeing if you can marshal your resources and get what you want. Okay? So, use them how you like, but this is what they do. All right, so let's see. Again, the specific enzymes. Let me say, talk about beta-glycosidases. Beta-glycosidase does the opposite that the glucoglycosyl transferase does. This enzyme puts sugars on. A beta-glycosidase takes sugars off. Okay, we'll talk about that more in detail on a different slide. But that's the enzyme I'm talking about. There's a family of those. One of them cleaves a terminal abiose. A different one would specifically recognize and cleave a terminal rhamnose, and so forth. Okay, so now moving away from a lot of the molecular biochemistry and plant storage and all that kind of stuff and structures and stuff. More on the concept now of looking at making white aromatic wines. Okay, and thinking about the title of the talk, where we're talking about extraction, preservation, all these different aspects of it. If we get to the point now where we start thinking about the winemaking process and how we can impact these molecules and processes. I've tried to put things together in somewhat of a coherent order, but it really doesn't matter. These are just different modules. Okay? So I'm not a viticulturist. I don't grow grapes. There's lots of things you can work with your viticulturist in your vineyard to optimize the quality of your grapes. And I think I still do pretty much believe that great wines are made in the vineyard, right? Because you really can't build a great wine out of average fruit, okay? But you can certainly take grapefruit and make a poor wine. All right, so with that in mind, I'm not bringing anything in from the vineyard. I'm not going to talk about how to moderate flavors in the vineyard because I'm not a viticulturist. I did stay at a Holiday Inn Express near Napa one time, but I'm still not going to talk about that. Okay, so right from the beginning, in terms of maximizing these flavor aroma compounds, besides what comes from the plant, you've got some modification of those molecules by the yeast. You've also have production of molecules by the yeast, fermentation esters, generation of enzymes to help modify precursors. All of these things are yeast driven. Okay, so I'm going to talk about yeast a little bit here. The first thing I want to talk about is yeast rehydration. Okay, and I always like to make a couple major points with this. First of all, yeast rehydration, okay, is not nutrition. It is not nutrition. Okay, rehydration products like GoFirm or Dynastar, they don't have nutrition in them. They have what's called survival factors. Okay, sterols, fatty acids, minerals, vitamins. All right, nutrition is carbon, nitrogen, things like that. You don't want to put that on your yeast when you're rehydrating them. Okay. And first, let me talk about why you don't want to put them on there. Then we'll talk about what these rehydration products do that's beneficial. Okay? We're going to talk about microbial physiology just real quick here. Okay? I like to try to keep it simple and talk about like happy yeast and unhappy yeast. Okay? Happy yeast are metabolically active. They're doing everything they're supposed to do. They're not stressed in any of the metabolic pathways, including primary metabolism, the uptake of sugar, glucose, glycolysis, the TCA cycle, generating all the energy they need for cellular process. That's primary metabolism. Okay? And I like to include the amino acid biosynthetic pathways as primary metabolism as well. Always in these cells, they're making proteins, and they're breaking proteins down. They're making enzymes to put in metabolic pathways to drive those, and then part of the regulatory aspect of that is they break them down, and they reuse the amino acids. So they always, with the amino acid biosynthetic pathways, it's primary metabolism, okay? And it uses nitrogen, it uses carbon. It pulls carbon skeletons out of glycolysis and feeds them into these pathways so it can do it. If I had the metabolic chart up here, you'd see it's just a bunch of lines all over the place. But for your yeast to be happy, all of those things have to have material flowing through them at the proper rates, okay? and the proper energy to drive these reactions. Cofactors a lot of times like ATP, okay? Things that you need to generate through glycolysis, TCA, generation of NAD, whatever, okay? When those things get sticky, the yeast gets unhappy. And when the yeast gets unhappy, it makes molecules we don't like making wine. Not the least of which, right off the top, hydrogen sulfide, a big one. Because in its environment, there's sulfur in the environment, and yeast take this sulfur up, much like they take up iron, okay? These are atoms that are very important for them to live, and they take them up when they're there. Whether they need them or not, they take them up, okay? If the metabolism isn't happy, they're not making cysteine, methionine, they're not putting that sulfur where it belongs, they're going to put it somewhere, okay? They make hydrogen sulfide, or they stick it on carbon skeletons that aren't cysteine and methionine, and it ends up as sulfides. Okay? Sulfides are all those stinky guys that you don't even know they're there sometimes because they can be in the form of disulfides. And then in reducing environment, they can be made back into mercaptans or thiols or single SH groups, and those are the guys that stink. Okay? So you can make a wine and it's not that bad, and then it evolves and gets stinky. Where does that stink come from? It wasn't in here. Well, a lot of it, if it's thiol driven, is because it was a disulfide that doesn't smell, or its threshold is 1,000 times higher. Okay? So happy yeast do things you want them to do. Happy yeast reproduce at a rate that gives you a good solid population, so that you can see as your fermentation is going at full speed, you're going to turn that sugar over into ethanol to dryness the way you want to. Okay? If the yeast are unhappy, they're not going to reproduce as much, you're going to get a lower population, it's going to turn that sugar over, it's going to get sluggish. All right? When things get sluggish or stuck, they're not all sluggish and stuck. Some of them, they're all still there, and they start doing things you don't like them to do, and you end up with stinky wine. Stinky wine is nobody's friend. So, all right, stinky wine, metabolism, happy metabolism. What does that have to do with rehydration? What we're seeing today, and this is from observations over the last 2 to 3 years, so it's relatively new. Let me take one step back. There's a chart that people use to make nitrogen additions, right? You've seen the table. It's got proposed YAN based and the potential alcohol or bricks. It's the Christian Butsky, Lyndon Bassan table they made like 20 years ago, looking at trying to find out cures for stuck and sluggish fermentations. And so they're looking at nitrogen supplementation using DAP. And so they found that if you have a DAP or a YAN level at a certain amount with a bricks level of a certain amount, that your ferments don't get stuck. And so that was really interesting findings. And nitrogen has been known as a supplement in must for a long time now, and it's helped a lot of fermentations. But it's more complicated than that. Okay? And what we've done is we've evolved from just DAP to complex nutrients that are mixtures of yeast extracts with organic nitrogen, amino acid content, as well as vitamins and minerals with DAP. Okay? And now there's organic nutrients that's all just yeast extracted nitrogen, has a high level of minerals, vitamins, but there's no DAP in it. There's no inorganic nitrogen in those organic products. Okay? Well, what we're finding is that these organic nitrogen compounds are used much more efficiently than DAP. Right now, the ratio sits at about 4 to 1. Okay? We're doing the work right now to kind of pin that down. But let's say, for instance, if you had a must come in at 90 and you wanted to get to 250, okay, you need to add 160 parts, all right? Which would mean 160 parts of YAN of DAP, okay? Or if you were going to use an organic nutrient, what you would use is 40 parts because it's used 4 times as efficiently. That's the difference. That's when you're looking at nutrition, okay? So let's go there. Let me take that nutrition aspect in organic versus inorganic and take it one step further and say, okay, what if the nitrogen supplementation wasn't the only thing, or maybe not even the most important thing, in terms of preventing stuck and sluggish fermentation and yeast performance in doing what we want it to do in fermentations? Okay? What we're finding is that when you rehydrate your yeast with a rehydration product, okay, that it's much less susceptible to nitrogen stress and stuck and sluggish fermentations. We have people that were adding 150 parts of YAN. Now they use Dynastart, they rehydrate, and they put in 20 parts, and they get the same performance. So rehydration makes a big difference in the overall performance of the yeast. Now, recycle back to when we first put these products out on the market and what we designed them to do, okay? Again, it was a lot of the causes for stuck and sluggish where people were looking at high alcohols. Okay, you get up to 14 and above. A lot of these yeasts that are commercial yeast were isolated from Europe. They make lower alcohol wines in Europe. You guys make lower alcohol wines here than my people do out in California. Okay, But at above 13.5%, 14% alcohol, it's very stressful on the yeast. So one of the things that these products provide is sterols and fatty acids that integrate into the membrane when you rehydrate. Okay? That gives the yeast strong performance all the way through the end of fermentation. They're much more resistant to ethanol. There's independent papers that have been published. It wasn't a Laffort paper. It was, I even forget who the guy was. But they published a paper on the specific sterol that gives high ethanol resistance in yeast. It's ergosterol. Okay? We have twice as much ergosterol in Dynastart as other rehydration products. So it's important, the fatty acids. Why is it important? Here's what this slide is about. Okay? Sterols, important in high alcohol ferments and completing your fermentation. Good membrane permeability and efficient transport systems. Okay? That's what we're talking about for efficiency and nitrogen use and all. Remember I talked about lipid bilayer? Well, here's a picture of a lipid bilayer. These are lipids, these long-chain fatty acids hanging off a triglyceride here. And these structures here are sterols. They're ring structures, they're kind of complex, okay? And what these sterols do is they give fluidity to the membrane, all right? And then think about, like, what I like to say is the difference between a membrane that's all fatty acids and one that has fatty acids and sterols. It's the difference between another fat, butter, okay? You got butter in the fridge, you got butter on the counter. Butter on the counter is kind of soft, it melts on your toast nice, the other one's hard, okay? That's the difference in membrane. Fluidity. And what does that mean? Embedded in the membrane are all these transport proteins. Okay, I don't have my complicated diagram today, but these things are everywhere in a three-dimensional fluid mosaic. Okay, in the lipid bilayer. These proteins have to have this distance from here to here, and this hydrophobic association against this transmembrane surface for these to work properly. Okay, so if you don't rehydrate the membrane and make it fluid and give these things the environment they need to be efficient and successful, your yeast metabolism is not going to be so successful. Likewise, if you put nutrients in your rehydration broth, okay, what you're going to do is flood the cell. Remember, these cells, when they're dehydrated, they're like little sponges. When you rehydrate them, they'll soak up everything in their environment. They'll pull these things into their membrane more readily. Okay, and they'll also have external things like minerals and vitamins will come inside. All right. Now, if you have nutrient in there, you flood the metabolic pathways with nutrients, and it chokes them, it clogs up their pathways. They regulate how much of what molecule they want in their pathways by bringing in substrates, breaking them down in specific proportions, regulating their genes to turn on pathways just for the amount of material they want to push through it. If you shove all this food down their throat, it takes them a while to gag it out. And that's why you'll get a sluggish start, or you'll get a population that's not as high as a population if you don't try to force nutrients into the rehydration stage. Okay, that's the big, it's not a nutrient reason. Okay, so you've got this membrane health. This creates happy yeast, and happy yeast don't make stinky things. Not only do they not make stinky things, they also produce more of what you want yeast to do. Okay? But we all have our favorite strains. I know everyone in here, you've done different trials, experiments. You found the strain that you like for your wine style and your variety and your vineyard. It works really well for you. Or if not, you're still looking for one. But yeast have different characteristics. The point for this is that they're not all the same. One size doesn't fit all, and they do different things. When you pick one out that does something you like, you want it to keep doing that, right? You don't want to make it unhappy so it stops giving you the good performance and starts doing bad things. So, more positives, less negatives. When you make your yeast happy, rehydration does this. You can see that you'll get higher levels of these compounds that you want, black versus white. This is increased levels. And to be honest with you, I don't know how much of this is yeast synthesis and how much is the yeast processing the precursors more efficiently. But whichever it is, trial after trial, when we measure control versus rehydration, the rehydration ferments have higher levels of the compounds you want. This goes back to the theme of the talk of optimizing the flavor aroma molecules in your wine. Okay? You want to grow your grapes the best you can, but you also want to do all the little steps throughout your fermentation and aging process to help maximize that. This is one. It's measurable. It's there. Okay? This is kind of a biased experiment. I'll go through this real quick. This was a must that had 160 parts of yam coming in, so it was kind of low. It's like 24 bricks. So it was a little bit low. Not totally low, but low. We didn't supplement it. Okay? We didn't supplement it. We fermented this guy with no supplementation, and we got all this hydrogen sulfide. This was an unhappy yeast in a low nitrogen environment. This same exact yeast, rehydrated in Dynastart, No nitrogen supplement, same amount of nitrogen, only made this much hydrogen sulfide. The rehydration makes a huge difference in the happiness of the yeast and the subsequent production of negative or positive molecules. Okay? These molecules are rotten egg flavors. They're similar to hydrogen sulfide, they're bad players. And you can see again, the regular one had this level and the rehydrated had this. This was challenged. You know, we challenged it with a low nitrogen environment. We wanted to see when yeast goes bad, you know, can we prevent that by doing the rehydration, and it was very successful. So that's one way you can maximize and minimize the negatives, maximize the positives. Okay, so real quick, yeast nutrition. I talked about this a lot already, so I'm not going to beat this up. I talked about making yeast happy, blah, 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 blah. I like to throw one thing out there when we talk about nutrition, and that's know your yeast. Know your yeast. Ask your vendor about your yeast. What is my yeast nitrogen demands? How much nitrogen does my yeast take to perform at its best? Okay? Because they're different. These are all Laffort yeast. Okay? And they're different in how much nitrogen is optimal for them. Some of them take as much as twice as much. Okay? So when you're planning your nutritional ads, do you take into consideration which yeast you're using and its needs? Usually not. Usually not. This information is in our catalog. It's easily accessible, and it's easily accessible from whoever you buy yeast from. They should be able to tell you. If you're interested enough to know and you want to gauge your nutritional program for your fermentation, it's a good place to start with some additional information about your yeast. Because remember, they make positive things, they make negative things. You want them to do more of one and less of the other. Treat them the way they want to be treated. Okay, here's a quick, the whole point of this slide is looking at the varietal aroma content and basically the complexity driven by yeast plus a rehydration. I don't like to use this word, rehydration product. I don't like to say nutrient for that. Versus a single yeast with no treatment versus two yeast, okay? A lot of people think, oh, I want a lot of yeast in there, I want a different yeast, I'm going to do a two yeast combo, I want to use, I want to do wild ferments because it's going to bring me more diversity. It's going to bring me more complexity, okay? I'm not going to say that's not ever true, but in this trial, it wasn't true. A properly rehydrated yeast, a happy yeast, makes more than a single yeast, and it makes more than a yeast combination. So again, making your yeast happy, and these fermentation esters down here, this one, the regular yeast, did better. But in this one, only the rehydrated made any. Here it was the highest, again, by a little, and here, this is a little bit more significant. But you can see whether you're talking fermentation esters or conversion of precursors, the proper rehydration of the yeast makes them a better performer in your fermentation than even two yeast.
When you did two yeast, were they using the rehydration nutrient?
No. The point in that was that the two different yeasts would bring more or more complex without the rehydration. No, the single yeast alone was not rehydrated, and the two yeast were not.
You just put them between two tanks or in one?
In one tank.
In one tank, two yeast?
Yeah, like a co-fermentation.
Were they rehydrated together or rehydrated separately?
I can't say that. I didn't do that experiment. I would imagine that they were rehydrated separately. Most of the time, I don't really know which yeast were used, but sometimes the protocol can be slightly different. I would imagine that they probably did them separately. I would. I mean, if it was my experiment, that's what I would. Okay. Here's a bit of a summary slide. Okay, so we talked about primary varietal aromas with oxypure. We didn't talk about that one. Thiols, terpenes, norisoprenoids. Secondary aromas, fermentation esters, right? The primary are usually things that come from the grape. Secondary, things that can be integrated esters, fusels. We didn't talk about fusels, just mentioned fermentation esters. But these are also compounds that are produced by yeast. And then what we call tertiary or aging aromas, and that's more your oak-driven furfurols or your yeasty aromas from, say, surly, not so much from active yeast metabolism, but from yeast autolysis and breakdown. And then, of course, oxidation. So, you have to ask yourself, which ones do I want predominantly in my wine? What can I do to reach that goal? And so with extracting and preserving precursors, you can have enzyme applications. We'll talk about fining can play a role, early juice fining. In preserving aroma precursors, reductive practices, okay? There's lots of things you can do just in your cellar practice. And I surveyed some winemakers to try to put a slide together for things. I wanted to be fair and not just talk about all this product application stuff, but winemaking stuff too. But it all boiled down to just, using CO2, using dry ice pellets in press pans, having CO2 blankets on wines, keeping tanks topped appropriately, watching your SO2 levels, keeping your free SO2 where it should be, 30 or wherever you're going, 30, 35 parts. In finished wines, cellar wines, oxygen management, minimizing transfers, okay, and minimizing filtrations. And I'll talk about some things about transfers and filtrations with applications as well. But those were the kind, that was pretty much it. Those were the practices for keeping things reductive, or reductive winemaking. That was pretty much what I heard. Other than some things like using antioxidants like glutathione or tannins. Some of the things we'll talk about going forward. But if it didn't fall under some of these applications, then I wanted to, there are ways to do things. Much like I said, like with the enzymes, with the beta glycosidases. Time and environment will also allow for the cleavage of those sugars. I'm not going to say that you have to buy an enzyme for that to happen. It happens all the time. But you can moderate it, you can drive it, you can take it where you want it to go and control it more with enzymes. Okay. Expressing, we talked about yeast rehydration, nutrients, oxygen, temperature. I didn't mention one thing about oxygen. I did mention sterols and rehydration products and getting these sterols in the yeast membranes. Yeast will make their own sterols, okay? But they need to do it in a very oxidative environment. So if you don't want to rehydrate, you want your yeast to have high sterols, you need to add a lot of oxygen in your must and get them going. Well, what does that do? How many other guys have you got in there that are actually aerobic that would love to have all that oxygen in your must? Okay, so there's microbial risk that's associated with using oxygen addition to get the same kind of sterile composition in your yeast membranes. So just something to keep in mind. Okay, then preserving during aging and bottling, temperature, again, cooler temperatures is good. Well, cooler temperatures can go both ways. Cooler temperatures also, with the solubility of oxygen, the colder it is, the higher the solubility of oxygen is. Okay, so you kind of got to balance the temperature control for keeping volatiles non-volatile, because they'll stay in solution better at colder temperatures. Also for just simple microbial control. Chilling things keeps microbial activity very, very low, or stops certain organisms from growing. And the last thing we do is operate in a sterile environment. Wineries, that's one of the good things. I'm mostly concerned with U.S. regulations, and I say FDA all the time, but really, we're not regulated by the food regulatory people. There's a reason for that, okay? And it's a good thing, because if we had to prescribe to all of the good manufacturing practices that food manufacturing facilities have to, we couldn't make wine anymore. It would be impossible. It couldn't happen. So, the good thing is that we have alcohol levels that are high enough that there's never, ever been a pathogen in a finished wine. That's why we'll never be regulated by the food people. It would kill our product, and there's no real safety reason to do it. Okay? All right, so that goes temperature, SO2, oxygen management, again, minimizing transfers, keeping tanks topped. So, I talked about vacuoles earlier, and I said I'd show you an electron micrograph of some vacuoles. Well, here you go. Here's a large central vacuole, and here's all these other guys in here. Here's some close-up pictures and others, 3 major vacuoles here. Here's these pictures. Here's all these nice little round spherical vacuoles that contain specific, you can see they're different colors. They contain different things. Okay, there's not one vacuole, there's lots of them. But the thing is, we have to get after these one way or another, right? That's where all of our goodies are. That's where our flavor aroma precursors are. That's where the anthocyanins are in our red grapes. But they're inside the cell. Okay? They're inside of what cells? Okay? Here's some pictures of some grape grapes. You can see that you've got an outer surface of compacted, flat, thick-walled cells, and then you've got a pulp inside that's these big, fat, bulbous cells that are full of sugar and acid. These guys, 80% of the aromas, except for Ruby Cab, all the color in red wines, okay? We got to get after what's in these guys out here. They've got a tough cell wall to work through. To get after all the stuff in here, mechanical processing, crushing, this is an oxidative process, even with CO2. Okay, so you're going to have some oxidation going on. You can't get away with that. And what I noticed when I first started in this industry back in '04, just kind of walking around taking everything in, because I wasn't a wine guy. When I started, I didn't even know that there was a malolactic fermentation. So there was a lot for me to learn. I did a lot of walking around and asking questions and sniffing things, and I said, God, this pomace smells wonderful. Why are we throwing all this good stuff away? There's so much stuff in here I would love to have in our final product. Why are we throwing it away? Well, I don't want to over-extract. I don't want to have too many soluble solids. I don't want to extract too many phenolics, blah, blah, blah. And I was like, well, so I developed this term called subtractive winemaking. And I was like, why don't? Because I'm an enzyme guy. And I'm like, man, let's use some enzymes. Let's get after all this stuff. This will be great. But no, we don't want solids. We don't want phenolics. We don't want bitter compounds, blah, blah, blah. So I was like, well, can't you pull all that out and then remove that other stuff without affecting the quality parameters you want in your wine? And in 2004, it was a little bit iffy. The solids management techniques that were being used at Gallup, they had a whole bank of decanter centrifuges. It was highly oxidative. Getting rid of solids was a big problem. They chose not to over-extract because the solids management to get the juice in fermentable shape, this is white winemaking, was was just too much of a burden. As far as the phenolics, the early finding of juice was not something people did then. It just wasn't a practice. Today it is. We have products we make specifically to do that do super well at pulling out the phenolics, caphtoric acid, fumaric acids, these things that are the really bitter compounds. You can pull those out selectively. And I'll talk about finding later. Okay, so what we're on here is trying to extract. So enzyme treatment, mechanical processing, crushing, skin contact enzymes. You can increase your yield of juice and your volatiles, but it can increase phenolics. But if you follow that with finding, then you get a positive without the negative. So let's take a look at what we're really up against. Here's a schematic of a cell wall. This is your basic cell wall. You've got cellulose microfibrils, you've got hemicellulose strings here, and you've got all this fill in between all of this stuff. Here's your cell membrane. Here's the outer surface. Okay? All this green stuff in here, this is pectin. Okay, it's kind of like a construction analogy I use. This is your rebar, these are your twist wires, and this is your concrete. Your pectin's your concrete. Okay? So you got to get after all of this. What you have to do is compromise this cell wall. We don't want to break it down. We don't want all these big polymers being like soluble solids and creating a big gooey mass in our wine, in our must. That's really hard to deal with. So we don't want to break this down completely. But we have to compromise this enough that this membrane becomes compromised and the guts of the cell leak out. When you compromise the integrity of the cell, everything breaks down pretty rapidly. All those lipid bilayers I mentioned for those different compartments, they tend to just fall apart because it takes maintenance to keep all this stuff together. So what you want to do is compromise this enough so that this breaks and the cellular contents are released. But you have to work through numerous layers of this. Remember how thick the cell wall is? Remember how many of those cells were stacked up? Okay, so we were thinking about how we were going to get after all that stuff that's inside the cells, in the vacuoles, and things like that. And basically, the way we get after grapes is with pectinase enzymes. Okay, we use the word pectinase. Pectinase is not one enzyme; it's a lot of enzymes. Okay, but real quick, let's just talk about what is pectin. Okay, pectin is the most complex carbohydrate, structural carbohydrate. Okay, cellulose is it's a linear chain of beta-1,4 linked glucose molecules. Hemicellulose is xylans and arabinans with minimal branching. And then you've got pectin that looks like this. You've got a backbone of repeating rhamnose and galactose molecules, galacturonic acid and rhamnose molecules. You've got another part of the backbone that is PG, polygalacturonic acid residues. And then you've got these intermittent regions like this with a lot of side chains branched on it. But in addition to this, you've got acetylation and you've got methylation on these sugars. And it's a very, very complex carbohydrate. Okay? So the point with the pectinase is that it's a lot of enzymes, and here's like the main 7 enzymes listed here and where they act. What you need to do with pectin is you've got to break down this molecular structure. When you break this down, the rest of what's left in the cell wall is like a sieve. It's like a screen. It's not going to protect the integrity of that membrane. Okay? You need this barrier of all this. As you can see, with all these carbohydrate chains and everything, this creates like a colloidal type matrix within that cell wall, and it eliminates a lot of things, and it creates like a turbo pressure. It's actually a pretty cool carbohydrate. Okay, but what you've got here is you've got three main enzymes. You've got polygalacturonase that cleaves between unmodified galacturonic acid residues. You've got a pectin lyase that cleaves between modified residues, between methylated galacturonic acid residues. And then you've got this fellow here, pectin methyl esterase, and this guy cleaves these methyl groups off. This guy works with this guy, because this guy, if you cleave these off, then this fellow can come in and break the backbone. Okay, this guy works alone. But these are the main activities for breaking this backbone chain. This RG guy, he'll break some of these over here, the difference between the R and G units as opposed to the PG areas, polygalacturonic acid areas. But these are your main enzyme activities, but just as important is knocking these side chains off. These endo-acting arabinogalacturonase, arabinogalacturonase, and these other guys that break the endo and exo, sorry. Exo-acting work from the ends of chains, endo break chains in the middle. Okay? Breaking them in the middle reduces viscosity rapidly. That's what we want to do. Because there's two things we're after by pectinase addition, right? We want to work on these cell walls and extract what we want to get out of them. If you're doing some, enzyme work on your whites, okay, or if you're using enzymes in reds, this is what you're trying to do. You're trying to get inside those cells of the skin. But also, whether you're with whites or reds, you need to break this pectin down because a lot of this pectin is soluble. It's solubilized. It comes when you break the pulp. There's a lot of soluble pectin in there. It's part of the ripening process of the fruit. When fruit gets softer, that's what's going on. There's internal enzymes that begin breaking these things down. Okay, and so juice has a lot of high molecular weight soluble pectins that creates viscosity that helps. It makes your solids management very difficult. Okay, that's why you add pectinases to your juice so that your solids will settle and you don't have to wait 6 days before you can rack your juice off and ferment. Okay, so there's really two purposes, but it gets back to the same thing. Breaking this main chain, reducing the molecular size of these pectins so that you can either extract from the cells or reduce the viscosity in your juice for solids management. So, and again, here's just an example. I'd like to talk a little bit about enzymes now. I'm going to try to keep this really brief because this is really what I've done for years and years. I can go on forever, but I won't. I'm going to make a simple point. Here's some product pictures, okay? The enzyme inside of those packages is different, okay? I know everybody thinks, oh yeah, it's enzyme. You put a different label on it, telling me it does a different thing and feed me the same crap, okay? That's not the way it works, okay? I work for a company that made enzymes, all right? And even in a field as restrictive as winemaking, where the regulations say you have to use an Aspergillus niger enzyme, okay? There's different strains of niger that make, you saw the different enzymes, the many, many different ones, okay? Different strains grow in different ways to produce the enzyme, can make different levels of those. You can have a high BPG product. You can have a high PL product. You can have a high arabinase product, okay? You can make these things through what you feed the bug when you do the fermentation and grow it to make the enzyme, okay? Versus the strain you use, okay? You can make difference. And then what we've done at Laffort, at our research winery in Bordeaux, is we've taken these compounds, these different enzyme products that have different levels of activity, and blended them for optimal use in winemaking. Okay? When I started in the industry in 2004, a lot of the winemakers didn't like the enzymes, and my job was to interface with all the vendors. I talked to each one of the vendors, what's your product, how do you make it, blah, 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 blah. And I asked them all, most of the time I asked the people questions they couldn't answer. They had to get back to the technical people. They had to have somebody fly out to talk to me because they couldn't answer my questions. They were salesmen. They weren't scientists. And I wanted to know the fundamentals of the product they were putting in front of me. Okay? But it comes down to, these things are really different. And when I started, a lot of the pectinase products were not made to be used in the wine industry. They were fruit processing. They were mostly apple. Okay? Because apples have been processed with enzymes for a long, long time. And so when it's got to be, hey, let's throw some of this against grapes too. Okay. Well, the pH is very different. The temperature range that we get to work in is very different. And the enzymes weren't very applicable. And so, this is one of the things that I've learned since joining Laffort, how we developed our enzyme products. And, we only work on wine. We don't work on anything else. We don't work for the yeast or bread companies, or, we don't make yeast for any other applications. All we do is make products for wine. So we weren't interested in an enzyme that worked on different fruits. We just wanted one that worked optimally in grapes and wine. So you blend these things together, you ask, what do you want them to do? So we separated the activities out. You can add an enzyme, excuse me, to extract from the skins on white fruit. You don't want to over-extract. You don't want to make a lot of soluble solids. You want to get those flavor aroma compounds out, okay? Extract. We blended these enzymes together to find the right amount of these activities to do that job very well. What if you're just looking for press yields? I want yield in my press. I want more juice, okay? It's kind of a different way to attack it. And we've got a different blend that does that. I guess I can share this with you. One of these is a high PL, this is a high PG. But it's got the other activities, but they're different, and they work differently. This is this enzyme CL. This is an enzyme that is for depectinization. Okay, you add this in your press pan or in your juice tank after pressing. You don't put this on the fruit. This is high PG and high PL. It's very active, and you wouldn't want to put that on your solids. Okay, there are differences inside of the packages, and they're tailored to do what we say they're supposed to do. And we've got the data to show that they do extract the positive measurables. They don't extract the things that you really aren't looking for, and that they will give you initial free-run juice yield gains, things of that sort. So enzyme use, you can definitely differentiate. All right, so enzymes, other enzymes. Beta-glycosidase, we've talked about this a lot already. I'm not going to belabor this anymore. It cleaves the sugar residues off of those precursor molecules so they become volatile. And you can see in a measurement of geraniol in this particular example, 2 grams per hectoliter, 5 grams per hectoliter. I'll say this about this product, beta-glycosidase. Our product is called Arome. Arome. That's the beta-glycosidase, all right? I always like to tell people, because when I first started in the industry, I thought this would be the best tool winemakers would ever have. Because you could do so much with the flavor aromas of wines that people, winemakers gotta love this. I asked the winemakers and they said, I hate that. I tried that enzyme, it sucks. I don't like it, made my wines bland and flabby and blah, all these other descriptors, they hated it. So I started asking them about how they used it, what size, where, and all this stuff. And they were using it like pectinase. And then a pectinase is like a hammer to pick. When you're going and you're doing a job, you're digging a tunnel, okay? It's a hammer, okay? This isn't. This is the velvet glove, all right? You should use this at half the recommended dosage initially. I tell everyone, bench trial this, always. Don't ever just use it, bench trial it, okay? Start off at half the recommended dose and work your way up in increments. Keep your wines at the same conditions as you'd have your wine in your cellar and monitor it, taste it every 2 to 3 days because it's going to evolve and change. And when your wine gets to where you want it to be, you can hit it with 1 pound per 1,000 of bentonite, drop the protein out, the enzyme, and it stops. Okay? Or if you go in with a really low dose, eventually the enzyme, it doesn't last forever in a wine environment. High alcohol, acid, enzymes don't last forever in those environments. Okay? So you can add a very low amount and just let it run its course. Or you can add for a certain amount for a certain time and drop it. Okay? But that's the way you use this enzyme. It's not like a pectinase. All right, so, and then beta-glucanase, I talked about that a little bit already. Our beta-glucanase is called extra lyse. And there's beta-glucans. Again, these are carbohydrate polymers, and they're very much like cellulose. But cellulose, like I said, is a beta-1,4 linked polymer. Beta-glucanases have beta-1,4, beta-1,3, beta-1,6. They have branches, okay? And beta-glucans are what ends up in your fruit when you get botrytis infection. Okay, and other fungal infections can also introduce beta-glucans, and you won't even know it throughout the whole vinification process until you go to filter. And when you go to filter and it doesn't filter, then usually what you do is you throw a beta-glucanase against that wine and it clears it up. Because the beta-glucanase has that specific enzyme activity, but it also has pectinase activity. It's got cellulose. We couldn't make an enzyme that's affordable for you to use that's purified. Okay? When we ferment these fungal strains to make these commercial enzymes, they make all of them because that's what they do. And we can't separate all those out without a large cost, and it's not feasible. So when you get a beta-glucanase, it's enriched in beta-glucanase enzyme, but it still has some pectinase activity, which is why it will break down all of those large carbohydrate polymers that cause your filtration problems. Okay? But the other thing this can be used for, and getting back to the flavor aroma context of what we're talking about, in the cell wall of Saccharomyces cerevisiae is beta-glucans. Okay, so if you're going to age your wine on lees, you want to do some surly aging, and you want to wait 9-10 months or whatever, then you can do that, and you can pretty much get what you need, I would suppose. But you can shorten your timelines and minimize your microbial risk of having wine sitting on lees in your cellar by adding some beta-glucanase and helping those yeast cells to lyse, so that they'll release their contents and give you those characters whether it's the manoprotein, 30 kD manoprotein that helps protein stability, or whether it's the 40 kD manoprotein that helps tartaric acid stability, or whether it's all the other manoproteins in that diverse mixture that lend mouthfeel characteristic, or whether it's the HSP12 breakdown peptide that supplies sweetness and roundness to wines. These are all things that we've learned through research, and I talked about in my Surly Science talk here last year. Those are compounds that come from Surly aging, that you can accelerate the extraction from these by use of a beta-glucanase. Okay, so beta-glycosidase enzyme targets, we talked about this already. The 4 different sugars, the di-glycosidic linkage to the volatile precursor, and splitting these things off with specific enzymes. So we've talked about that. All right, everybody's favorite, oxidation. So I like to start off when I talk about oxidation. It's kind of chemistry, oxidation, chemistry. I'm not a chemist. So a lot of this stuff is, there's people that know a lot more about it than me, but to try to get familiar with it, I always start off with the real basics with oxidation. What is oxidation? Okay? Well, if you've got a hydroxyl group, say alcohol, like ethanol, you can oxidize it to a carbonyl, which is an aldehyde, which would look like this. This is an oxidation step between ethanol and acetaldehyde. So you can see what's going on here. This guy is gone. This becomes electron dense here. You can oxidize this further into a carboxylic acid, acetic acid. So that's this is the real oxidation cycle that we're most concerned with, because what we have is hydroxyls are the fresh component on our flavor aroma precursors. Where is that? Right there. Okay? These are the guys that we need to protect from oxidation to aldehydes, or even further on to carboxylic acids. So again, oxidation, these arrows that represent oxidation, what is that? What's behind that arrow? What does that really mean? Okay? Where does most of that come from in wine? Okay? Mostly it comes from the Fenton reaction. Anyone ever heard of the Fenton reaction? Yeah. Well, it's mediated by iron, okay, with copper as a cofactor, and it goes on all the time. You can't keep iron out of wine. It's like impossible. You could chelate the iron with specific chelators, but they're really toxic compounds. You wouldn't want to drink that wine after you use that. So there's no way to get rid of it. And so what it does, it's iron dependent, and it's got a peroxide reaction product. It takes water and it oxidizes it to this radical, hydrogen peroxide. And then this guy goes all over the place, interacting with all these different molecules, oxidizing them. Which is why it's so hard to generate a metric for oxidative damage in wine, because this is a highly reactive intermediate, okay? And the products of its reaction are spread all throughout the molecular sphere of the wine. So you can't really pick one thing and say, this level of this indicates this is 20% oxidized. You can't do that, okay? That was the basis of some research that Andy Waterhouse started about 3 years ago that led to some really cool discoveries that I'll talk about in a second. But oxidative damage is hard to detect. Okay, so Fenton reaction, here's your Fenton reaction, here's your generation of hydrogen peroxide, okay? This is a model that was put forth by a fellow named John Danilovich out of the UK. And between this fellow's work and Andy Waterhouse's recent work, they've really shed some light on oxidative processes in wine that we're right on the edge of finding some really good treatments or ways to deal with oxidation in wine in a very productive way because of the work that these guys did. And they looked at things like the interaction of antioxidants with Fenton reaction components, and they looked at things like the cooperativity of these molecules and the kinetics of the reaction, meaning which ones react first and how fast do they react in relation to each other. So really cool work, and I'm not going to present all that today. That's a lot of chemistry. But in the meantime, just say, phenolics are antioxidants. They can play a role in this whole redox cycle. SO2 is an antioxidant. It can play a role in this redox cycle, okay? And they work together. They don't work into, you can't say, well, I don't care about phenolics. I'm just going to load it up with SO2. That's not so cool. You can't say, I'm just going to use a bunch of tannin. I don't want any SO2 in my wine. That's not going to work optimally for you either. And here's the proof right here. Here's what Oenology did. He's got all these oxygen level in the wine and removal of that oxygen by the addition of these antioxidants, okay? When you added these tannins alone, it didn't work out so well, okay? But when you use SO2 plus the tannins in conjunction, it worked out pretty well. See, they work together. That's what helped him propose this interactive model. So, just redox tower, different reactivities or levels of reactivity. The point I make with this is not any chemistry. I base this. SO2 in wine, where does it come from? Where does SO2 come from in your wine? How does it get in your wine in the first place? My head says it right there. Yeast produce SO2. They produce low levels of SO2 and winemaker addition. You guys make decisions. You put SO2 in, that's cool. What else? What about tannins? Tannins come from grapes, they come from oak, and they can also come from winemaker addition. Okay? Here's your tannins. Glutathione. Well, we'll talk a lot about glutathione, about what it is. Where does it come from? Glutathione is a component of grapes. It comes in and from the vineyard in pretty high amounts in your grapes. Okay? It's also in yeast. Okay? And now you can also add it through winemaker addition. So, let's take a look real quick. I just want to say about tannins. All right? They're a very diverse molecular family of molecules. They have a lot of double bonds. They have a lot of oxygens. These guys are electron sinks. So they're like buffers for electrons, which makes them a good redox partner. Okay? Whereas a regular buffer works with hydrogen ions, right, and buffers pH. Okay? These guys buffer electrons and they buffer redox. So that's why they're good antioxidants. Okay, so tannins again. I'll mention this. White winemaking. Okay, I'm not going to talk about VR Supra, VR Color so much right now, but say tannin gallicule. This is a tannin. It's from a tree gall. All right, and it's a really good antioxidant. It's also a good protein binder. But tannin gallicule additions at the crusher provides antioxidation protection for and you see where the polyphenolics come into play in terms of the cycling with SO2. It helps the effectiveness of reductive winemaking. I mean, if you want to try to blanket things with CO2 and minimize the oxygen ingress, if you throw some stuff like this in there as well, then if the little bit that gets through, you've got a buffer system against it. So it's not one thing you can do. I'm not going to sit up here and say, if you do this, your oxidation problems are gone. What I'm going to say is oxidation can happen from the minute you break the grape open all the way until you put the wine in the bottle and beyond. Okay? And it takes a lot of little steps all the way along and doing what you can do in order to minimize that. Okay? Tannin addition is a good way to buffer against redox, against oxidation. Okay, protein binding affinity gallicule again. I'll say especially in varietals like Sauvignon Blanc or Pinot Gris that are really high protein, it takes a lot of bentonite later to heat stabilize those wines. The earlier you get after that, the better off you'll be. Your precursors are conjugated here. You're not going to blow them off. You're not going to run them off. They're still going to be there later. But after you've made the wine and things have been modified, the more you find and work and rat and remove and filter to get your wine back out of your lees, you're going to lose those volatiles. Okay, so the earlier you get after this stuff, the better you're going to do for your aroma molecules in your wine. And then again, tannin addition before wine movements. There's people I know that they'll take like product Cortannin, which is oak tannin. They'll do a simple like a 10 ppm add to a tank the day before they're going to move it, because you can't not pick up oxygen. The connections, it's just, you're always going to pick up a part or so. So to minimize the oxidation, you'll throw 10 parts of Cortannin in before they do the movement. Okay, glutathione, another antioxidant. We talked a little bit about tannins. Tannin can help. Okay, how many people have heard of glutathione? Know what glutathione is? Right. It's kind of hit the human health news in the couple of years. It's an antioxidant. It's a really potent intracellular antioxidant. It's a tripeptide. It's 3 amino acids. Okay, this is cysteine, this is glutamic acid, and this is it's 3 amino acids, but it's made enzymatically inside of cells. It's not made on a ribosome like most peptides are. Or proteins. So, basically, okay, the point to make here is just adding glutathione won't do you a whole lot of good, especially if you add it really early, because, like I said, there's always oxygen that gets picked up, and once glutathione gets oxidized, it can't be recycled. It needs an enzyme, glutathione reductase. It can be recycled inside of cells, which is why the strategy with glutathione is to add it to the ferment so that the yeast take it up. And you enrich the yeast in glutathione, and then during the death phase of autolysis, they give that back to the wine, and you end up with protected glutathione in the reduced wine. It's less of an oxidative environment once the fermentation is complete. With all, when you finish the fermentation and you put all the CO2 is bubbled, all the oxygen out of the wine and all, then it's a much more reduced environment than in early fermentation. So that's why we tend to, or whatever, there's instructions in this a little bit later. Okay, so biosynthesis, again, I mentioned the enzymes, I won't burden you with that. You add this product at the first third of fermentation so that it's not oxidized by quinones and must. Now, there's always phenolics in the must, and it's kind of an oxidative process early in the fermentation. Okay? And then as the ferment gets going, it becomes more and more reductive in the fermentation. So, as a peptide, especially with the sulfhydryl group, this is subject to oxidation. So you want to keep it away from this early oxidative portion of the fermentation. So you put it in, it will be metabolized by the yeast all the way to the end of alcoholic fermentation and then released during autolysis. So for optimal protection, measure and correct the YAN of the juice. Why would the YAN be important for the use of an antioxidant peptide like this? Correct your YAN, make your yeast happy with their nitrogen. If they're not happy with their nitrogen level, they're going to cannibalize this and eat it. They're going to break it down and use it for nitrogen instead of keeping it as an antioxidant. Okay? So that's why the nutrition is important. Protect the juice against oxidation, again, through normal reductive winemaking practices. And then maintain the conditions during aging. Keep your tanks topped, keep your SO2 levels up, minimize transfers, things of that sort. CO2 blanketing, things of that sort. Okay, so real quick before I talk about glutathione in the juice, I mentioned there's a lot of glutathione in grapes when they come in from the vineyard. Not so much in juice and not so much in wine. I'm guessing that probably nobody here has seen like an HPLC profile, a phenolic profile from juice. If you do, you've all seen like a chromatographic profile where you've got big peaks like this and little spikes like that. There's a big hump in juice phenolic profiling, and it's GRP, grape reaction product. That's caphtoric acid plus the glutathione. Okay, so rapidly when you start to crush grapes, it's an oxidative environment, and there's a lot of caphtoric acid, and it binds really rapidly with glutathione. So glutathione is really high in the fruit, but you're not going to get that glutathione throughout the winemaking process because of the early reaction with the caphtoric acid, plus all the oxidative steps that occur in early processing. So you end up with things like this, low values like this, and then in the corresponding wine you can see that the yeast through autolysis have given a little bit of a bump. Okay? Now glutathione in juice is proportional to the initial YAN in juice. Okay? If the YAN is high, glutathione is usually high. But it doesn't really matter because the glutathione in juice is lost rapidly anyway. So, great glutathione can be lost rapidly. Good alcoholic fermentation nutrition, carbon-nitrogen balance, I mentioned that earlier already. You can supplement the glutathione in the yeast with a timely nutritional addition. Timely about 1/3 of the way through the ferment. Okay? And then what we found is 20 parts. You need 20 parts of glutathione in the finished wine for optimal protection. Okay? And what do we mean by optimal? How can we challenge oxidative protection? Here's the example right here. You've got these two compounds that are waxy and honey. They're oxidized flavors. And you can see that without at the end of alcoholic fermentation, without any glutathione additions, you have this level. After 9 months, you have this level. This is confusing me. Oh, okay, these are the two compounds, I'm sorry. So you can see that at the end of alcoholic fermentation, these guys are here. The control after 9 months, they've increased at this level. They've oxidized more of these initial compounds, oxidized in these here. And with the bio-aroma, it's been a lot less. You've got that much less and that much less in these two compounds. So with an appropriate addition of the glutathione, you get the redox protection, and these oxidative molecules don't form. Okay, boy, to get through that, I better study that one next time. Okay, so, and again, preservation of positive molecules. Here's the elimination of negatives. Here's the preservation of positives. Okay, here you've got your 4 MMPs, and this one doesn't affect very much the 3MH. You've got a bit of an increase, and the 3MHA, a significant increase. This is preservation of these molecules. This would be a reduction because of oxidation in the untreated. I'm not going to worry about that further. So this is, I really should put this one first, I think. Here's the trial we did, glutathione, 20 parts, bottling, on the thials, oxidative aromas of Sauvignon Blanc, 4 years. 4 years, that's a long time for Sauvignon Blanc to hang around. Usually people get that out and get sold, but just the same. You can see that with these negative compounds, waxy and honey, that in the control, they're at these levels, and in the treated, they're dropped down to these levels. So it's kept these from forming. And then the same thing, these fruity aromas, again, significantly higher values. Another slide exhibiting the protection of from controlled to bioaroma treated wines. Okay, I've talked about most of this stuff already. The only thing I haven't really talked about much is wine finding. I just kind of mentioned it in passing. Let me address this with a couple of quick slides. I talked about early finding and juice finding. This is something that we're doing a lot of work on now, and it hasn't been a big part of what a lot of people do in terms of finding of you wait and you find your white wines, you don't find your juice so much. But so there's a way to get rid of you can oxidize your phenolics in your press, okay, or in your juice. You can use an oxidative process on your juice in order to let these phenolics oxidize to quinones, react with each other, and precipitate out. They form all, you can brown your juice, and then that stuff will fall out. But what also happens then is you can oxidize other compounds, and you'll also lose all of your glutathione content through oxidative. But how could you get rid of all of these phenolics that are here in, say, an inert press? This is protected from oxidation. How can you get rid of all these phenolics without losing oxidatively either your aroma compounds or have oxidative damage or lose glutathione? You can do this, and then you can treat your juice with a reducing environment, and then with the phenolics you can find them out. Ferulic, fumaric, and caffeic acids, these are the main phenolics from white pressing, and you can get these out with, we've got a product we call Polymusk, and it's a PVPP, bentonite, and isinglass combination product. We also have another product that's casein and PVPP called Polylact. It would help if I knew all the product names, but those are finding agents for use in juice to drop these phenolics out without having oxidative damage. And again, like I said, the sooner you get after your juice, the better off you are. Finding it when it becomes wine can be difficult. Here's an example, just pictorially. Here's the juice untreated, controlled, browning goes on. Here's this wine, this is after fermentation, sorry. And then you ferment the wine and then treat it with Polymusk, and you can see it doesn't really pull all these phenolics out very well. But with the juice treated with Polylac or the juice treated with Polymusk and then fermented, you can see you don't have any of the phenolic browning. So this is just a treatment, a dosage level treatment, and a reduction in 420, which is a yellow color. So you can see with the more you treat with the finding agent, the more you reduce that color. And then this is an example of the guys you're actually pulling out, the caphtoric and the catechins, and you can see that with the different dosages from 50 to 100 to 120 parts, how much of these phenolics that you pull out so that they don't end up as brown. Okay, finally, summary. Good news for everyone, huh? Okay, so we talked about the volatile compounds, the different families, where they are from, and what they look like. We talked about the flavor aroma compounds, where they're produced, and how they're stored, and what that means as far as us how we want to get after them and get them into our wines. And then extraction and preservation through a number of different means: enzymes, juice finding, fermentation, tannin use, glutathione antioxidant, yeast strain, and the importance of nutrition in beta-glycosidase maturation enzymes. So that's just a list of all the products that we spoke about for the different purposes. And with that, thank you for your time and patience, and hopefully try to answer any questions you might have.
Has anybody got any questions?
Anyone have any questions?
Yes. To use some of these foaming agents, I don't find any of my juices currently. I didn't actually pay much attention because it does say in the book they're recommended for musk or for wine. Is there a value of pre-fining it as juice versus post-fining it as wine, I guess? Are we doing the same thing? Would we still need to do these fining treatments on the wine if we did it on the juice, I guess? Two questions.
Okay. Fining on the juice, first of all, it's an aqueous environment. It's got a different level of interaction between these components without all the alcohol in it. Okay? The interactions are a little bit better with the fining agents and the tannins in the non-alcoholic media. So that's one reason to do it on the juice and not wait to do it on the wine. The other is really the concept of stripping the wine of the volatile aromas afterwards versus with the juice. Any volatile compounds you have in your juice to begin with before you start your ferment, they're gone. Because when you ferment, all that CO2 bubbling through that as you make that wine is blowing all those things off. Okay? So you really don't have to worry about stripping in your juice. But once your wine is made, they begin, the yeast will make the enzymes, that will release some of these aroma compounds. The alcohol, the acid environment, as it sits in the cellar, you don't find it the day after you get done making it, but you'll start to liberate the compounds that are going to be the basis for your finished wine. And when you go to do additional fining later, you'll strip some of those out through the process. Now, will it replace fining of wine? No. Because during the fermentation process, you're going to develop some fine solids that are going to have to come out. You're also going to have, usually in a white wine, especially some of the higher protein varietals, you're going to need to bentonite treat to make them heat stable. Unless you're doing a completely unfined, unfiltered style. But most of the time, you want your wine to be heat stable, so that's a bentonite addition. And in varietals like Sauv Blanc or like I said, Pinot Gris, it can be pretty high protein, and sometimes you have to do 2 or 3 additions. Even with bench trialing, you'll get a value, say 7 pounds. You'll do 7 pounds, you'll retest the wine, and it's not stable yet. Because you underestimate because there's so much there. So knocking some of that down early can help you do a single fining step later instead of having to do multiple. Did that help?
Excellent.
I wonder if I rambled too far off the top.
Any other questions?
Yeah, I have a question. We just have, with regard to enzyme use, in your experience, with juices clarified through a centrifuge, what do you suggest? Like any particular difference that uses, respect just like a cold temperature clarification or a form of filtration of juices, just for centrifuge. Well, you mentioned before your experience with Gallo and all those things, so those of time, any particular change difference?
Well, there's three important characteristics for enzyme use in winemaking that we can control, and that's time, temperature, and dosage. Now, the temperature is a very narrow control because we like to keep the juice cold because we don't sterilize. So that one is narrow. But the time and process is where you can really allow things. And you have to balance your time and process versus your dosage. Okay? Which is why I recommend things like come with a press dose at like a minimal dose. All right? And get your press yield. And then worry about depectinization and how rapid with a product like CL in the tank. I don't think one product fits everything. And it wouldn't be strange to me to use low levels of the extract to get more varietal character out. Come back with a little bit of the press in order to maximize your yield. And then, depending on your time and process, if you've got a couple of days where you could let your must settle, then you might not need to add any more enzyme. Or you might need to hit it with a certain dose of CL afterwards to complete the depectinization and move to your next step. So timing is very important, and dosage. Those are really where your control handles are.
Anybody else? I've got one actually. What was the difference on the sensory properties of the wine between the DAP, when you added DAP, and organic nitrogen? Did you do sensory at all on it as well?
We always do sensory evaluations on everything, but I can't say that I was not part of that panel. One of the things that we do notice is that with the organic additions versus the DAP additions is that you do get a more complex series of flavor aroma molecules made. They say we do at Sarco, our lab in France and in Spain, we do flavor aroma analyses, we do the fermentation esters, we do terpenes, we do the noise products too. We do, that's not a common analysis in the U.S. or here in Canada, but I send samples back over there. And also, what that also means, a lot of the data that I receive from our research team over there at the University of Bordeaux has had this type of analysis done. And so the best I can say, since I haven't had hands-on, I didn't do those particular experiments, but looking at the data sets that I've seen, with the organic nutrients, you get a more complex mixture of flavor aroma molecules.
Any other questions, anybody? Well, I'd like to present you with a special edition, much sought after, CCOVI corkscrew.
Thank you very much.
You can't all have one, is it? So thank you very much indeed. Thank you to Laffort. Thank you also to Vines and Vintages. And thank you very much also to CCOVI for sponsoring today. And thank you very much, Peter, for your time.